Hi everyone, my name is Luo Pei. I am a postgraduate student from Shanghai Jiao Tong University in China. It's my great pleasure to give a presentation of my work in this conference. And the title is The Disordered and Highly Correlated Oral and Gut Microbiomes in Gastric Cancer Patients. Gastric cancer is a major health problem in many countries. It has a huge geographical heterogeneity. A study on 2020 reported that the Asia-Pacific region had the highest incidence rate, especially China had nearly half of the global incident cases. And according to the statistics reported by National Cancer Center of China, gastric cancer is one of the top 5 common cancers, and it showed a higher incidence in males compared to females. Gastric cancer is a multifactorial disease. Some factors such as geographical locations, daily diets, genetic inheritance, and stimulus exposure are reported to be associated with the risk of GC. And infection with Helicobacter pylori is the strongest factor. It will cause chronic gastritis and then induce the carcinogenesis. So, HP has been listed as the Class 1 carcinogen by WHO. In the stomach, except for HP, it is now known that there are many other bacterial species. The composition and interaction of the gastric microbiota varies in different stages of gastric cancer development. And it was found that the mice transplanted with gastric microbiota from GC patients could replicate the premalignant lesions, which indicated that other commensal bacteria also contributed to gastric carcinogenesis. Besides gastric microbiota, oral and gut microbiota were also found altered in gastric cancer progression, respectively. And many research suggested that poor oral health and specific species enrichment in oral cavity are associated with the high risk of GC, and this may be mediated by the translocation of oral pathogens, because the colonization of oral bacteria on gastric mucosa was found to be common among GC patients. And oral species were more abundant on gastric mucosa of GC compared with any other developmental stages. There is a research published on gastroenterology in February of this year. They found enrichments of Streptococcus aeruginosus and Streptococcus constellatus in both gastric tumor and feces of GC patients. However, the source of these two Streptococcus species are unclear due to the lack of corresponding oral samples. Because the oral cavity, as a reservoir of bacterial pathogens, may play an important role in gastric intestinal diseases through oral bacterial translocation. For example, the oral bacteria Klebsiella pneumonia from IBD patients can colonize the colon and trigger a strong inflammatory response. And the Fusobacteria nucleatum, which is a very famous pathogen in CRC, colorectal cancer. It was initially found to be enriched in feces of CRC patients, and it would promote colonic inflammation and tumor proliferation. We therefore wanted to investigate the oral and gut microbial differences between GC patients and healthy individuals, as well as the correlation of the oral and gut microbiota among the individuals of two groups. Besides, we tried to identify the potential translocated oral bacteria associated with gastric cancer. So we included 45 gastric cancer patients and 43 healthy individuals, followed by the inclusion and exclusion criteria listed here. And for the GC patients, we collected the mouthwashes, cancerous tissues, non-cancerous tissues, and feces. For the healthy individuals, access to their gastric mucosa is not allowed, so only mouthwashes and feces. The subjects' recruitment and samples collection were achieved through Shanghai Changzhen Hospital. All the samples' DNA were extracted and sequenced the V3V4 region of 16SR gene, and we analyzed the microbial data based on ASVs identified by DADA2 plugin. We found that the oral microbial community structure of GC patients is significantly different from healthy individuals. After filtering the ASVs present in more than 10% of oral samples, we got 393 ASVs, and these ASVs were clustered into 46 co-abundance groups. We call it CAGs. Among these CAGs, there are 5 CAGs showed significant lower abundance in GC patients' oral cavity, and 3 of them are positively correlated. The ASVs in these CAGs are mainly from the genus Streptococcus, Leptotrichia, Megasphere, Fusobacteria, Treponema. Besides, GC patients' oral microbial network had lower complexity, including less nodes, edges, average degree, connectivity, modularity. And in GC oral cavity, there was a unique module comprising 2 negatively correlated groups of bacteria. We see that Fusobacterium, Porphyromonas, Neisseria, and Prevotella are negatively correlated with the Streptococcus, Legionella, and Leptotrichia. Next is the gut microbiota. We see the gut microbiota of GC patients was also different from healthy individuals. In GC gut, there are 6 CAGs significantly enriched, and they have a strong positive correlation with each other. These enriched ASVs are mainly from Streptococcus, Lactobacillus, Klebsiella, and Enterobacter. And it's worth noting that many species from Streptococcus, Klebsiella, and Enterobacter are proved to be opportunistic pathogens. In addition, there are 6 CAGs reduced in GC gut, containing ASVs from Anaerostypes, Faecalibacterium, Ruminococcus, Butyricoccus, Eubacteria, and Bacteroides, and many species of them are widely reported as short-chain fatty acid-producing bacteria. Then we analyzed the correlation between oral and gut microbiota. Among GC patients, we found that the diversity and richness of gut microbiota are positively correlated with the oral microbiota. We even see the significant similarity of oral and gut microbial community structures based on Procrustes analysis. However, there is no such correlation among healthy individuals. Besides, GC patients have significantly smaller distance between oral and gut microbiota compared with NC. Then, we analyzed the ASVs present simultaneously in oral cavity and feces of at least one individual. We call them OF shared ASVs for short. There are total 221 OF shared ASVs, and 6 of them have significantly different oral gut co-occurrence rate based on chi-square test. Interestingly, the 6 ASVs are all having higher oral gut co-occurrence rate in GC patients. Actually, we can see they are widespread in the mouths of all individuals, no matter GC or NC groups, and their abundance in oral cavity of 2 groups have no obvious difference. However, they have higher prevalence and abundance in the gut among GC patients. Among the 5 OF shared ASVs, 3 of them are Streptococcus. Others are Peptostreptococcus germina. From the heat map, we can further see the 5 key OF shared ASVs distribution in oral cavity and feces among GC and NC individuals. They are oral residents in almost all of the individuals, but they appeared more and enriched in the gut of GC patients, which indicate that these 5 oral bacteria translocated more to gut among GC patients. And when we mark the OF shared ASVs as the yellow and red color, we found that the enriched CAGs in GC gut are mainly consisted of OF shared ASVs, except the CAG2, and they are positively correlated. Then, all of the gastric tissues were classified into 2 clusters based on DMM model. We found that the tissue samples in cluster 1 contain low abundance of HP, and in cluster 2 contain high abundance of HP. And GC patients whose 2 kinds of tissues, both in cluster 1 or both in cluster 2, were classified as low HPGC or high HPGC patients. We found that the high HPGC patients had higher gastric bacterial loads, lower microbial richness and diversity, and different gastric microbial community structure and composition compared with the low HPGC patients. However, we didn't see significant difference between the cancerous tissue and non-cancerous tissue of the 2 kinds of GC patients. Besides, we found the 5 key OF shared ASVs were also present in the gastric tissues, especially the ASV of Streptococcus mitis. It is most abundant and widespread. By the way, the oral and gut microbiota of high HPGC and low HPGC patients didn't show significant difference in richness or diversity or the community structure. So, here gives the brief summary. The oral and gut microbiota of GC were significantly altered, and among gastric cancer patients, the gut microbiota was highly correlative with oral microbiota. And oral microbiota were more likely translocated downward to gut among GC patients. More importantly, we identified 5 translocated oral bacteria may be associated with gastric cancer. And actually, 3 of the key oral bacteria were already isolated. And then we want to know that, are they passers which just pass through the stomach, or are they the drivers which promote the gastric cancer progression? We will conduct the experiments in vitro and in vivo to further classify the role of these bacteria. That's all for my presentation. Thank you very much for your kind attention.